Okay, thank you for your introduction. So today I'm going to talk about a tool we developed. It's called ImmunoMatch, which is useful to illuminate the design of antibody heavy and light chain pairs using deep learning approaches and structural analysis. So now, especially after COVID, a lot of people are familiar with the concept of antibody. So antibody is produced by B cells, and it functions by using the V region to bind to the antigen invented into human body. For this reason, a lot of research they focus on how the V region bind to the antigen. But if we take a closer look of the structure of antibody, we will realize that actually the antibody is a complex. It has two heavy chains and two light chains. And all these chains and domains, they interact with each other to form the final antibody, which function in our body. So we have this question of how does a heavy chain choose its light chain partner? This question seems simple, but it's actually not easy to answer. Because if we think about how the antibody is generated, it will go through this process called VDG recombination. So in our germline, we have different gene segments for both VDG genes. And all these segments would do a recombine with each other to form the final segment, which will lead to the production of the antibody. So this process will repeat for both heavy and light chain. And as a result, you will have 10 to the 6 different heavy and light chain combinations. And it's difficult to determine which light chain partner this heavy chain want to choose to produce the final effective antibody. Moreover, during the development of the B cell, one of the precursor B cell is called pro-B cell. It will first try to produce a heavy chain and then produce a light chain. This is where the VDG recombination occurs. So after this, if the antibody, they can form a stable heavy and light chain pairs. They cannot form a stable heavy and light chain pairs, they cannot gain the survival signal. So this B cell will die. Although you can also have a second chance, they will go through this process called light chain rearrangement and to see if they are able to produce a stable heavy and light chain pair. So if the B cell are able to produce a stable pair, they will go through another check to see if they react against your self-antigen. And if no, they will continue their development to become a mature B cell, which produce the antibody. If we think about this question from a structural perspective, so as you can see that this is a H domain, this is a Val domain, and this is the antigen it binds to. So if we zoom into the interface between the heavy and light chain, we will see that actually there are a lot of radios buried at this interface. We don't know how this would influence the compatibility between the VH and VL. And then we realized that actually people already start to investigate this problem of compatibility, but actually it's not in the context of antibody, it's in the context of human social life. This tool is called Tinder. So in case some of you are not familiar with this app, it is an app for people to use to search for your soulmate. So the Tinder app will try to capture the features of the users and then to determine which two people can form a pair or not. So if we borrow this concept of the Tinder into the pairing of the heavy and light chain, we can develop a tool which we call ImmunoMatch. So basically, the idea is that if we use the same analogy, so we have this heavy chain, and then it has a lot of light chain candidates. For example, this candidate is a light chain with a very flexible loops. And we have another light chain which has a fancy helix at the top. So our goal is that we want to use our tool to learn the features of the heavy light chain to determine which heavy light chain pairs can form a more stable match. So this idea sounds interesting, but how do we achieve this? We want to use some machine learning models to be able to learn the features of the pair heavy light chains. So in order to do this, we need to have a lot of data to fit into the model. So our training data is acquired from the single cell sequencing technique. So basically the idea is that in this technique, for each cell, you attach the barcode in front of the heavy light chain so that we know that this heavy chain pair with this light chain. So in the end, we collect all these like 6 published data set from 3 different papers. And then we sample equal amount of antibodies from like each data set to form our training data set. And then we use the 90% for training and 10% for testing. So these are positive examples in our training data set, but how about the negative ones? Because the model needs to also learn the feature of the unpaired high-value light chains. So it's not easy to get the negative examples, because if they cannot form a stable high-value light chain pairs, as I explained previously, this B cell will be like killed, and then you cannot be able to sample it from the blood. So what we did is that we generate the negative cases, which we call the pseudo-negatives. So you can see that you have the two heavy light chain pairs you sample from the single cell sequencing technique. So these are labeled as paired examples in our training data set. And then for the negative ones, we simply just like switch the light chain partners from randomly 2 paired heavy light chains. And then this will be our unpaired heavy light chains in our training data set. So this generation process meaning that in our training data, we have equal amount of paired and unpaired sequences. So the training data set is not paired. It's not like a bias. It's balanced. And then we want to try different like machine learning architecture to see if there are any signal of the heavy light chain pairing. So first, we just like use a very simple input information. We just encode all the V and J gene usage and input it into like a basic machine learning model architecture, such as logistic regression and random forest. So as you can see that the F1 score is around 0.5, which means that the model cannot be able to distinguish the paired heavy light chains from the unpaired ones. We think probably it's because the model doesn't have enough information to learn this. Because if you take a closer look of the structure of the antibody, we can see that actually between the heavy light chain interface, there are a lot of radios of the CDR3 sequences interact with each other. So this time we add another feature, which is the encoded CDR3 sequences. For logistic regression, you can see that the performance is still not good. But the random forest, the F1 score is slightly higher than 0.5. It's not ideal, but it means that there are some signals there, and there are hope that we can further improve this model to achieve our final goal. So what we did in the end is that because nowadays people like adopt different language model in solving biological questions. So we just like take one pre-trained antibody language model called antibody2. And then we feed in our like concatenated heavy light chain sequences into this model to fine-tune the model and to allow the model to learn what is paired antibody heavy light chains, what is unpaired heavy light chains. So the model will generate a score indicating the identity of the pairing of the antibody. And we want to evaluate the performance of ImmunoMatch. So you can see that from this ROC curve, the area under the curve is around 0.75, which means that the model can distinguish between the paired and unpaired ones. And all these measurements is not bad. So our model is quite successful in distinguish between the paired and unpaired ones. And if you compare the performance of ImmunoMatch with all the basic like baseline models we showed you previously, we can see that there is a significant improvement of the F1 score and the ROC AOC value. So our ImmunoMatch can distinguish the paired antibodies from the unpaired antibodies. It looks nice, but what is the use of it? So we were thinking, can we use this tool to optimize the pairing for the therapeutic antibodies? In order to investigate, we take a popular therapeutic antibody trastuzumab, which is broadly applied in the treatment of breast cancer, to use that as a case study. So what we did is that we take the sequence of the trastuzumab, and then we try to mutate each position to all the 20 amino acid, and then we score the mutation by the pairing score generated by ImmunoMatch. So as a result, you have this huge heat map. So these x-axis are different radios of the trastuzumab, and then the y-axis are different mutations it will mutate to. And if we like gather all the mutation scores at each position, we will acquire this scatter plot. So the axis as a heavy chain radius and the y-axis as a pairing score. The red lines are the score for the wild type. So we can see that actually you have a lot of space to optimize the heavy light chain pairing around the CDR loops, and also some as a free mode region. So we want to map this scatter plot onto the antibody structure. So what we did is that we take all the mean value and standard deviation for each residue positions, and then we have this plot. So you can see that if the color is white, it means that it doesn't actually influence the pairing. Also, if the width of the structure is very small, it means that all the mutations at this position tends to have a similar effect. So this is the interface of the heavy light chain. And we can see that for the mutations away from this interface, it actually doesn't really influence the pairing, and this is what we expect. So from this mutational scanning, we are able to identify the point mutations which could optimize the pairing problem, but we are still not be able to identify which radial pairs are important to play a role in the heavy light chain pairing question. So what we did is that we want to extract the attention matrix during the training of our ImmunoMatch. So what is attention? For example, in the language model, for example, if you want to use it in the human language model, and you want to use it to translate an English sentence to a French sentence, you get this attention matrix. I don't understand any French, but from this attention matrix, I can still be able to make some guess. For example, the English word really probably means this French word rhythm after the translation. So if we borrow this concept into the heavy light chain pairing problem, we are able to identify the radial pairs which are important in the pairing problem. And this is the attention matrix we get in the end. The y-axis are the heavy chain radius, and the x-axis are the light chain radius. The lighter color means that these radials are as CDR loops. So we are able to identify some radial pairs which are close to the CDR regions and also some around the free mode regions. And if we map all these radial pairs onto the structure, we can see that actually, although the model is trained on antibody sequence, it will still be able to capture some structure information. Like they think that the radials as interface between the heavy light chain are important for the pairing. So in summary, our ImmunoMatch tool are able to make the prediction of the antibody pairing identity. And also, this pairing identity will be able to help us to understand the formation of the antibody repertoire. We can also potentially use it to design better heavy light chain pairs in therapeutic antibody design. So in the end, I want to thank a lot of people for this study. I want to thank my supervisor, Franca, and also the postdoc in our lab, Joseph. They give me a lot of nice and useful suggestions in this project, and also a lot of ideas for these slides. Yeah, that's it. Thank you.
Thank you. Thank you, Dongjun. We have time for questions.
So, excellent talk. I wonder whose ideas the Tinder slides were, but I think on a more scientific note, if you go back a bit to your attention head representation of stuff, so it doesn't immediately strike me as something that's very doable, right? So this matrix is not immediately available from the way that you think about attention heads in my head. So what did you have to do to get here?
So I think basically, because this is after the fine-tuning of the language model, so we just get all the attention matrix out. And then because they have multiple heads and multiple layers, so we think that the information in the last layer might be more informative in the pairing. And then we just like scan through all the attention metrics from each hat to say which one like looks more informative for the pairing, because some of them is like what we kind of expect is like the diagonal. So it's like something it's to itself. And this one, it kind of like give us some ideas about the pairing.
I guess where I'm going is that you're using an embedding per residue, right? So your attention heads are focused on a per residue basis, or like, you didn't really explain the back end of this.
So like because in the attention matrix for each, so it has the length of the sequence length, and for each like sequence length, you will have for each value you will have information about the like the model's weight at this residue. So if this is what you mean.
So you're encoding every residue. You're not doing any sliding windows. You're just encoding
Yes.
every residue.
Yes.
Okay. Thank you.
Any other questions? I have one. So just, I'm not familiar with this field. How big is the space in terms of the number of pairings that are, I mean, is this something that's, is there a large level of conservation in these regions? Is this all like highly mutated at these interfaces?
Yeah, so I think, so first of all, during the formation of the antibody, you first go through this VDG recombination, which already gives you a lot of like combinations of the heavy light chain. And because the goal of the B cell is want to produce the antibody, which can bind to the antigen better. So it will also go through some like mutations to like improve your affinity to antigen, which will make this problem more complicated. So in the end, the result will be like more combinations than the like number I showed you previously.
All right, thank you.
Thank you.
Sorry, just one more quickly. Just quickly, just out of curiosity, because you use the data set K paired sequences for your
Yeah.
fine tuning. Any reason not to use, let's say, the observed antibody space paired ones, which have around 10 times more, around 1 million sequences, just out of curiosity?
Yes, because we when we like investigate this problem, we want to use a healthy antibody from healthy people, and then in the OES you also have like antibodies which has disease. So we didn't take the entire OES. We just used like the healthy antibody repertoires. And I think some of the data sets is already in OES.
Thank you.